I thank the organizers of PMD Family Support Meeting and PMD Foundation for giving me this great opportunity to share some of our recent work with you. My name is Ken Inoue. I'm a physician scientist working on PMD, and associated diseases. Today, I'm going to talk about our recent effort on developing molecular therapies for PMD using gene therapy technology. This is my COI disclosure. Okay, before starting my talk, let me introduce myself. I was born in Tokyo 2 years before the last Tokyo Olympic, although I remember nothing about it. After finishing my medical school, I was trained as a psychiatrist, and I am still a psychiatrist to see patients once in a while. I was working for Professor Jim Lupsky in Baylor College of Medicine, where I learned a lot about molecular genetics of genomic disorders. After spending almost 7 years in Houston, Texas, I came back to Tokyo and get a position at my current research institute to continue my recent research on PMD. For those who don't know me, I have been working on PMD and associated disorders quite a while, and these are some of my historical publications. Throughout these works, Professor Hitoshi Osaka, now in Jiji Medical University, has been a great partner of mine, mainly helping me on clinical evaluation studies. This picture was also taken at ASHG 2018 at San Diego with my wife Kimiko and Carlos from PMD Foundation. This picture shows the main target cells for the treatment of PMD, the oligodendrocytes, which generate myelin. Myelin consists of layers of thin membranes wrapping around the neural axons. By electron microscopy, good myelin looks like a dense circle wrapping around the axons. This slide introduces PMD and its causative gene PLP1, but I think I can skip the details for today's audience. This slide may be a little bit complicated, but it explains the different PLP mutations, associated clinical phenotypes, and distinct cellular and molecular mechanisms as to how mutations cause hypomyelination. Here, the genomic duplication is the most common form of mutations, representing 60 to 70% of the patients. The duplication presumably causes overexpression of PLP1, which has been thought to impair the cholesterol processing in the cells, but the exact mechanism still remains unclear. Nevertheless, too much PLP1 has deleterious effect on myelinating oligodendrocytes. We've been also working on point mutation as well, but I'm not going to talk about it today. Maybe next time if you have a chance. As I mentioned, PLP1 duplication leads to its overexpression, and thus downregulation of PLP1 may serve as a primary treatment for PMD. Now, there are a couple of choices in technology to employ, thanks to recent progression in the molecular genetics tools. Genome editing is the most recent and the powerful technology that enables permanent change on the nucleotides of one's genomic DNA as you wish. Antisense synthetic oligos are another potential tool for PLP1 downregulation, and this will be another topic of this session by Grace and Paul. But the approach we decided to take is the gene therapy technology using adeno-associated virus, AAV, for the PLP1 suppression, and more specifically, developing an AAV vector harboring an artificial microRNA specifically targeting PLP1 in oligodendrocyte. This slide shows a schematic strategy of our gene therapy of PMD. With a single copy of PLP1, oligodendrocyte can nicely myelinate axons of neurons. When PLP1 is duplicated, the messenger RNAs transcribed from the PLP1 gene are presumably doubled in amount, which eventually result in failure to myelinate axons, which is termed hypomyelination. To fix this condition, we aim to reduce the amount of PLP1 messenger RNA using artificial microRNA. MicroRNA are short RNA of 20 to 25 base pair in length, More than 1,000 microRNAs are present in human genome, and they inhibitory regulate gene expression by binding target messenger RNA. We have designed a new microRNA, thereby termed artificial, which specifically binds to PLP1 gene and disrupts its mRNA. For the delivery of this artificial microRNA, we employed an AAV vector, which is known to be the best viral vector for gene therapy of neurological diseases for its safety and efficacy. As you see in the previous slide, the idea is relatively simple, but there have been quite a bit of challenges to develop the ideal therapeutic tool. Again, I'm not getting to the details, but we developed an AAV artificial mRNA that target PLP1 in oligodendrocyte-specific manner, as illustrated here. Now, Venus is a green fluorescent protein marker which can visualize the cells that are infected by the AAV virus. From here, I quickly go through the actual data to show how efficient our AAV therapy is using wild-type and PMD model mice. These two pictures shows mouse brains after injection of the AAV vector show highly efficient oligodendrocyte-specific expression that our AAV vector has achieved. We got more than 70% of oligodendrocyte being infected by the AAV where it was injected. These are enlarged pictures of the brain, again showing oligodendrocyte-specific expression of Venus overlapping with oligodendrocyte marker, GST-Pi, but not with other cell type marker for neurons, astrocyte, or microglia. These data shows how our artificial mRNA can suppress the PLP1 gene expression. Again, green color shows the infected cells, magenta color shows the PLP1 protein. Upper panels are the negative control AAV doing nothing on PLP, and the lower panel shows the AAV harboring PLP1 miRNA. As you can see, PLP1 expression is clearly decreased in the therapeutic AAV by 50%. This was also true at the mRNA level, measured by quantitative PCR by 60% reduction. Now we get the tool. We next ask if this AAV can rescue the phenotype of animal model for PL duplication, which is PLP1 transgenic mice, shown in here, which overexpresses PLP1. It has been known that these mice showed severe hypomyelination in their brain, just like PMD patient, but they can only survive about a month. We directly injected AAV into the brain of TG mice at postnatal day 10 and see the survival and body weight curves. The AAV therapy significantly extended the lifespan of TG mice and also rescue the body weight phenotype as well. Normally, PLP1 is present in the myelin fiber as seen in the wild-type mice, but the brain of PLP1 TG mice, PLP1 accumulate or abnormally accumulate in the cytoplasm of oligodendrocyte. The AAV mRNA treatment rescued the cellular pathology almost completely. Accordingly, the number of oligodendrocyte, which is reduced in the TG mice, was rescued in the AAV microRNA therapy. These are electron microscopic pictures of myelin in the mouse brain. On your left, you see wild-type mice with normal myelin nicely wrapping around axons. And in the middle, TG mice shows massive loss of myelin compared to the wild-type. But if you treat these mice with AAV microRNA, you can see the myelin is nicely rescued with increased number of myelinated axons when we count them. By measuring the axis of each myelin fibers, we also confirmed that the thickness of myelin is rescued. Finally, we examined the effect of the AAV microRNA treatment on the motor function of the mice. Here in the middle, this test measures the time of mice to fall from the bar. TG mice fall in around 20 seconds, but the treated TG mice can hold the bar more than twice as long. On the right shows the limb slip test, which measures the motor coordination of the mice. TG mice shows more slipping while they were walking. They walked on the wire mesh than the wild-type mice, but this phenotype was rescued by the AAV microRNA treatment. All these data suggest that the AAV microRNA treatment also improves the motor function of the TG mice. Altogether, we show the proof of concept that gene-specific artificial microRNA delivered by AAV vector may serve as a new therapeutic strategy for PMD caused by PLP1 genomic duplication. For future clinical application of this gene suppression therapy for PMD, further technological development for broader and more efficient delivery of AAV is required. So what's next? We still need to develop better technologies and tools to bring this AAV therapy in clinic. These include optimization of AAV serotype, administration method, and improved therapeutic efficacy. But these are all doable. We also need to clear the safety issue and availability for vector production, which need to collaborate with a pharmaceutical company. Finally, this acknowledges all my long-term collaborators who's been helping me a lot to make our project moving forward to the right direction. That's all I have today. Thank you very much for your attention.